Hi everyone, in this video I'd like to go through the questions from the RE on genetic expression and mutations. To start off, you are given in question 1 a diagram that shows a part of a DNA molecule. I hope by now you can identify the key features. For example, number 1 most definitely has to be the sugar. So immediately, right, some tips if you have gone through MCQ with you before is I want you to be able to rule out options really fast. See that number 1 has to be the sugar. So that means you're only left with option B and C as the correct answer. A and D you can immediately cancel out. Often MCQs are like that. You can always very quickly rule out half the options. Option 2, you require a phosphate group. Okay, so now we have 3 possible options. B and C still hold. Now it comes to 3 and 4. 3 and 4, we know that they are nucleotide bases and we have A, T, C, and G. The question is, how do we know whether it's CG or if it's AT? Well, actually, I think for this particular question, if you look at the options provided between B and C, A definitely cannot bind with, interact with C, right? It has to interact with a T. C can interact with a G. So therefore, your best answer is C. Next, let's go to question 2. The diagram illustrates the relationship between DNA, gene, and chromosomes. Which option is correct? So let's see if we can rule out options. First, if you look at this giant structure over here, it most certainly is a chromosome. So therefore, I only have A and B to work with. You can cancel out C and D. Let's look at Q next. When we look at Q, you're still referring to this double-stranded structure, and that has to be a DNA. So we're left with option A as the answer. Gene is a sequence of DNA. So this is what a gene is. Let's go to question 3. Question 3 shows the events that occur in translation as listed below. There are various statements, 1 to 6, but they are not in the correct sequence. Which shows the correct order of events? First and foremost, I notice that there's one option that says translation ends when the stop codon is reached. And I like to think that that's probably how you want to end the sequence, right? So that means option A and option C is likely the answer. Because if we look at what 1 is, 1 says the previous tRNA leaves the ribosome. Okay, I don't think that's how translation ends. Okay, so now that we are left with A and C to work with, let's look at the options. First, we begin with 6. 6 is tRNA with methionine binds to the start codon. Now we either diverge. We either go for 3 or 5. Okay, let's look at the two options. In 3, ribosome moves towards the 3' end of the mRNA. In 5, the tRNA that recognizes the next codon binds to it. So which happens first? Actually, 5 happens first. If I were to draw out the RNA for you, and this is codon, okay, let's say this is codon 1, this is codon 2, codon 3. What you'll find is that the ribosome will bind to it, and then ribosome actually comes with like 3 slots where you can have tRNAs binding to. Usually this is where the start codon is when it first binds to. So you have the first tRNA with its amino acid. The second tRNA will then come in, alright, and then after which peptide bond will need to be formed, which is option 2 over here. Ribosome catalyzes a peptide bond formation. So a peptide bond will form between the two. Okay, then after the ribosome moves towards the 3' end of the mRNA. So this entire ribosome structure will then move down, and as it moves down, this will then go to this slot over here. This tRNA will go to this slot over here, which then leaves room for this area here for a new tRNA to come in. Right, so the option is C. Let's go to question 4. Which option correctly describes the effect of nonsense mutation? The idea of a nonsense mutation is that a stop codon is produced. So the option is already a quick one. This has to be D. If we replace one amino acid for another amino acid, as says in option C, this is a missense mutation. Increase the length of amino acid. Okay, usually nonsense code mutations result in a shorter amino acid sequence. No effect. Okay, that's very unlikely. If you have a stop codon suddenly introduced, there has to be an effect. So the option is D. We'll now go to the structured questions for RE 6.1.